Assalamualaikum. My name is Nur Farahin and I will present about the spread of disease between rubber and also PADDY. So for this slides, I will present about general introduction about this topic. Plant disease, an impairment of the normal state of a plant that interrupts or modifies its vital functions. All species of plants, wild and cultivated alike, are subject to disease. Pathogenic organisms such as fungi, bacteria, virus, protozoa, insects, and parasitic plants are mostly responsible for infectious plant illness. Infectious plant diseases have become an increasingly important element affecting agricultural productivity and economic efficiency as agriculture has progressed. Next, I will present about PADDY. For PADDY introductions, one of the most destroying viral infections impacting rice is crop rice stripe disease. This disease is caused by the rice stripe virus, which is consistently transmitted by the small brown plant hopper and transmitted to the following generation by transviral transmission. Next, I will talk about one objective about PADDY disease is to control of the vector insect throughout the crop's early growth period to enable the growth of sufficient number of healthy panicles. As the point shows on these slides, these are the summary of method for prevent the disease on PADDY. So, on 11 July, the panicle initiation stage, 8 and 9 August, flowering stage, and 4 and 5 September, just before harvest, all plants in the survey plot in Field A were examined for the presence of sick plants. During the survey in early August, we also tally the number of total infected and also healthy panicles on the fixed point survey plants. On the same dates as the Field A surveys, we evaluate disease around 8 plants in Field B. So in early August, Field A counted the total of infected and also the healthy panicles of the fixed point survey plants in Field B. So plants were considered infected if they had typical rice stripe disease symptoms including wilted young leaves and also malformed panicles. We move to result and also discussion. To analyze disease damage, we sample both infected and also healthy plants from the fixed point survey plants in Fields A and also Field B. We picked disease plants around the survey plants since we were unable to obtain enough disease fixed point survey plants for analysis. So the table below displays the brown rice productions, number of brown rice kernels, 1,000 kernel wide, number of panicles, and number of healthy panicles for each disease onset period, and also survey field for these plants.
Assalamualaikum. My name is Nur Atika Sufia binti Madlan. For the introduction of disease for rubber farming is presently threatened by fungi-induced plant disease, particularly leaf fall disease, a serious disease produced by Phytophthora species. On 2015, this disease was discovered on RRIM 600 rubber clone in southern Thailand, causing 75% defoliation and significant yield loss of up to 15%. Under humid condition, water soak patch on leaf emerged, followed by a small drop of coagulated latex. Under good condition, each spot gradually expand and merge to form huge and irregular rooting patch and the leaflet fall off. For the objective of this disease to modest inhalation of disease severity and evaluate in the laboratory and under greenhouse condition. Summary of method is Trichoderma isolate. The endophytic Trichoderma species were obtained from the leaf of healthy rubber plant aged 5 to 10 years. They were dried, cut into 0.5 cm pieces, surface disinfected by dipping into 70% ethanol for 3 minutes, wash with soda water for 5 minutes, soak in 10% NaOCl solution for 3 minutes, and finally wash with soda water for 5 minutes. The treat plant sample were dried with sterile tissue paper before being placed on potato dextrose agar or known as PDA that containing 0.1% streptozin sulfate to avoid bacterial contamination. With 4 sample pieces per plate. For 5 days, all plates were included at room temperature 28 Celsius with a 12/12 light or dark cycle. The trichoderma mycelia that developed from the sample were placed in PDA sun. There were 59 endophytic trichoderma strains detected in all. Result and discussion for this method is 12 trichoderma strains out of 59 strains display effective antagonistic activity. They were fast-growing strains and could grow over the colonies of P. palmivora within 4 days, which is in Figure 1. Then they were selected and classified as 5 species: T. asperillum, T. hematum, T. harzianum, T. longibrachium, and T. virens. The current study found that endoptic trichoderma species isolate from the rubber leaf that were effective in reducing Phytophthora leaf disease. To the best of our knowledge, this is the first investigation on the use of endophytic trichoderma species in the control of Phytophthora palmivora leaf disease under greenhouse and field circumstances. For the conclusion, controlling rice strip disease necessitated integrated pest management which incorporates pesticide-based insect vector control, planting of RSV-resistant cultivars, and paddy field management aimed to break the disease infection cycle. Meanwhile, endophytic trichoderma species have been shown to be possible new biocontrol agents against rubber disease, and they can be developed as alternative fungicide or used in conjunction with suitable fungicide. So that's all from our group. Thank you.